I really feel honored to present some of my research work, where my teachers are here, and august galaxy of scientists, and above all, my beloved students. So this work was conducted at Iowa State University when I was there for my PhD. And this is focusing on, can we use feathers? The big question that we have is, can we use feathers as a non-invasive tool to detect avian influenza virus from commercial poultry? So that is the main thing we are going to talk for the next 10 minutes. We know that avian influenza virus can be divided into high path and low path, and we know that high path viruses can cause lots and lots of mortality, even up to 100%. And one such outbreak, such epidemic, animal epidemic, which was the largest animal epidemic at US in those days, was 2014-2015, when this outbreak affected 21 states and caused loss of up to or even over 50 million chickens. Most of these outbreaks were in the Midwestern states of Iowa and Minnesota. And therefore, my advisor and I started to think about and discuss there are some gaps in the National Poultry Improvement Plan of the US, because sampling is done only for limited number of times and on limited number of samples per flock, no matter how big the flock size is. So we started to think about the environmental samples such as feces, water, feeding sources, feathers, air, and we found out that feathers are very limitedly used. They are non-invasive, convenient to collect, and also meet the animal welfare requirements. So we said that why don't we use the feathers for detection of avian influenza virus from commercial poultry? And this is all the study is all about. So what we did was we collected from various farms located in Iowa, we collected feather samples, and farms we selected were non-negative for avian influenza virus at that time. We spiked the feathers with the virus that we had. This virus was of low pathogenic origin and came from the migratory wild birds that we have the virus in our repository. So we spiked the virus onto the feathers and we kept the feathers at different temperatures for different times. And then we wanted to see if the virus titer is decreasing in those times or not. So we call that a half-life study. And half-life, we can define that the number of the virus particles that are decreasing over time. So that is what it is. We used the RT-PCR on these spike virus and we detected the RNA from these virus spike samples. Later on, we found out that what is the limit of detection of our PCR, and then we also use statistical analysis, logistic regression for finding out that how our spike feathers are doing in terms of virus detection. We found out that PCR-based estimates were close to, and here we can see, close to what we expected. Once we spike almost the same amount of virus, same type of virus we could detect. However, the PCR could only detect up to 10 to the power 3 EID50 per ml, but not lower than that. With a consistent 100% detection, only 10 to the power 3 EID50 per ml was detected by this. This is the same data set over 48 and 72 hours, and we can see this consistent trend that only 10 to the power 3 EID50 per ml was detected in the feathers. Again, coming back to the half-life, this is the half-life estimates in hours, and within the parentheses, we have the confidence limits, and this is when we are not able to detect. The upper bound is undetectable, and we call this is infinity. So the shorter the half-life, the more decay of the virus. So we can see that more decay of the virus in the DMEM, but in the feathers, there is virus is there, can be detected for longer period of time. This is very important because once we are shipping feathers, once we collect, once we are shipping them at the room temperature, then we can see that on feathers, the virus is able to survive for a longer period of time. And that has a diagnostic value in terms of shipping the samples to the diagnostic facility and then later on detection by the diagnostic facility. We concluded that loose or shed feathers can be used not just for avian influenza virus, but for other, these data can be extrapolated for other RNA viruses or other respiratory viruses. My work was funded by Ag Industry Center, and I was on a Fulbright scholarship, and that was also funded by the United States Department of State. My lab manager, Dr. Baoqing Gao, he helped me in execution of the experiments. Yuko Sato helped collecting my committee member, collecting samples from poultry facilities. And Dr. Bogger, my committee member, helped us in conducting the experiments. Anna Walk helped me in the statistical analysis. And Dr. Yoon provided insight, oversight, and everything, my advisor. This is the paper that came out of this study. I have a few copies of the paper with me. If you are interested, you can get those from me during the lunch break. Thank you very much for your attention. I'll be happy to take any questions perhaps in the lunch break. Thank you.